Good day. Thank you for the opportunity to present our work entitled Genome-Wide Association Study of African Esophageal Squamous Cell Carcinoma. My name is Chen and I'm presenting this work on behalf of the ERICA SA study. Esophageal cancer is the 8th most common cancer in the world and can be classified as either adenocarcinoma or squamous cell carcinoma. In Africa, there is a particularly high burden of disease for the squamous cell subtype of esophageal cancer, with high incidence areas in eastern and southern Africa. Our aim was to test whether common genetic variations in the South African black population contributes to the risk of esophageal squamous cell cancer. And to test this hypothesis, we conducted a genome-wide association study for ESCC cases and population controls from the South African black population. Our cohort comprised of 1,686 histologically confirmed ESCC cases recruited from 3 sites in South Africa and 3,217 population controls recruited by the Origin study. We performed the genome typing using the Illumina H3 Africa custom microarray, and we performed genotype imputation using a Sanger imputation service with the African Genome Resources Panel. After data quality control, we performed linear mixed model regression using 14.4 million SNPs. One of our biggest challenges was the presence of complex population substructures within our cohort. We used a combination of 3 methods to control for the population substructure to ultimately reduce false positive signals driven by population differences rather than the disease phenotype. Our final model contained 5 principal components and a kinship matrix as covariates. The diagram on the top right is our quantile-to-quantile plot with a genomic inflation factor of 1,01, indicating good control population structure. We identified strong signal on chromosome 9 that reached genome-wide significance, and the signal on chromosome 2 near the genome-wide significant threshold, both shown by the red arrows in this Manhattan plot. Our top hit on chromosome 9 is located near the FAM120A gene. FAM120A encodes an RNA-binding protein that protects cells from oxidative stress-induced apoptosis by activation of SERC kinases. Our second top signal on chromosome 2 is located within the intronic regions of the MYO1B gene. Interestingly, there appear to be 2 independent signals originating from the same locus. The top SNPs in this window are African-specific and monomorphic or very rare in other populations. Further to this, we conducted a trans-ethnic meta-analysis of our GWAS data with a published ESCC GWAS conducted in the Chinese population. We identified 4.85 million overlapping SNPs from both studies for the meta-analysis, and identified 3 genome-wide significant signals located on chromosome 9, chromosome 10, and chromosome 22, and 1 near genome-wide significant signal on chromosome 2. The signals marked by the green arrows were largely driven by the African GWAS, and the signals marked by the red arrows were largely driven by the Chinese GWAS. The meta-analysis provided added insights to ESCC genetics. For example, the FAM120A gene signal, although not significant in the Chinese GWAS, when combined with African GWAS, it provided a significant boost to the signal. Additionally, the signal at the PLCU1 locus was entirely driven by the Chinese GWAS, suggesting that the PLCU1 locus has little effects for ESCC in this African population. In conclusion, this is the first GWAS of ESCC in an African population that detected strong association signals upstream of FAM120A on chromosome 9 and at MYO1B on chromosome 2 for SNPs unique to African populations. Meta-analysis of our African GWAS with ESCC cases and controls from the Chinese population detected genome-wide significant associations at FAM120A on chromosome 9, PLCU1 on chromosome 10, and CHEK2 on chromosome 22. The study confirms the presence of a genetic contribution to ESCC risk in an African population and demonstrates the power of trans-ethnic meta-analysis to identify common or distinct risk loci in populations of diverse ancestry. Lastly, I would like to acknowledge and thank all the participants, team members, and collaborators who have made this work possible. Thank you.